Hi and welcome. I'm Natalie Caldell, and I'd like to walk you through a short lesson about standardization and measurement and talk about how they fit into the engineering of biology. And again, as we've done in other lessons in BioBuilder, I'm going to start with some historical examples from other engineering disciplines and ones that hopefully you'll find interesting and that you're familiar with. I think for many of us, we're familiar with the fact that standards seem to be non-standard a lot of the time. Here's a comic strip from not too long ago from XKCD, which talks about how, well, two folks here are talking where there happen to be 14 competing standards around something, and one of the stick figures says that 14, that's ridiculous. We need to develop one universal standard that covers everyone's use cases. The girl says, yeah. And so soon enough, of course, there are 15 competing standards. And some of the cases that you might be familiar with for competing standards, English versus metric measurements are competing standards. For those a little bit older, maybe they remember Betamax versus VHS as video recording technologies. And then even further back, as hard as it is to believe, the standard of alternating versus direct current was quite contested early on, with Edison being quite vocal and demonstrative about the standard he wanted to promulgate. So I have a picture on this slide that I like. It's a beautiful train, a new train that was from the Mason Machine Works in Taunton, Massachusetts, built in 1873. And what you may notice about it is that this beautiful train is actually sitting on a flat car, and it was needed, the flat car was needed in order to deliver this train for the Atlantic, Mississippi, and Ohio Railroad, because the train that was built in Taunton would not fit on the tracks to which it was being delivered. So it needed the gauge of the train that was built was different than the gauge of the train tracks where it was being built, and so it needed a flatbed to be delivered properly. And so you can see there's just a ton of wasted energy and a lack of usefulness if there are no standards that are adhered to, and a lack of utility when people can't agree on simple aspects of what they're building. In biology, certainly standardization is a somewhat new idea about how DNA, pieces of DNA, would get moved around in a standardized form. The Registry of Standard Biological Parts is one repository for standardized parts of DNA. It's a standard that we'll walk through in some detail. It is certainly not the only standard. There are other ways that DNA is being standardized and different aspects that are being considered, but we'll walk through this example from the Registry of Standard Biological Parts. It is a standard that is promulgated and supported by the BioBricks Foundation, so that's another place to look if you want to think about standardization and what it means for sharing of materials. The kind of DNA part that is found at the Registry of Standard Biological Parts is called the BioBrick. BBA stands for a BioBrick Series A. It is a way of standardizing a biological part. The part, again, is any DNA-encoded biological function that we as humans have defined. And the way these parts are standardized is that they are all preceded by what's called a prefix. This prefix has two restriction sites in it, echo R1 and XBA1, XBA. It also has a suffix, these parts. The suffix has two restriction sites, SPI1 and PST1. And what those enable is the ability to put these pieces of DNA together in a standardized scheme. The standardization of the assembly of DNA has been picked up in different venues. One of them is New England Biolabs. You can actually at least as of now, look in their catalog and find BioBrick assembly help and a kit for assembling pieces of DNA that conform to this BioBrick standard. And the way the assembly scheme works is that if you have a part of DNA, a particular part that you're interested in, we'll call it the upstream part, and here it's shown in blue, that part will be flanked with EchoR1 XBA on the upstream side, SPE1 and PST on the downstream side. If what you'd like to do is attach that part to a second part, here shown in green, and that part has the same flanking sequences, then the method is to simply cut the upstream part with EchoR1 and SPE, cut the downstream part with XBA and PST, and ligate them into a backbone, a destination plasmid that has been cut with EchoR1 and PST. And the composite part that you see at the bottom is hopefully what you've been trying to build, but what you can notice about it is that it itself, this composite part, conforms to the standard of biological parts. It has EchoR1 and XBA in the upstream prefix. It has SPE and PST as the downstream suffix, and in between the two parts, the blue and the green part, is a mixed site. It's a mixed site because the SPE that was the overhang on the upstream part and the XBA, which was the 5' overhang on the downstream part, when you ligate those two together, the overhang for those two restriction sites is identical. But when they are ligated, they do not regenerate either the SPE or the XBA site. And so you have a mixed site in the middle, and the prefix and suffix sites are maintained as unique sites for further cloning. So one thing that this scheme requires, well, one thing it enables is for you now to not have to wonder how are you going to piece two pieces of DNA together. It takes away the sort of ad hoc cloning strategies that you need to come up with in order to build small pieces of DNA and assemble constructs. One thing that it requires of you to get this benefit, though, is that you have to refine the pieces of DNA that are between the prefix and the suffix so that they do not have any of the restriction sites that are used for cloning. So this part, BBA B0034, can't have EchoR1, XBA, SPE, or PST within its coding sequence. And one of the examples that I think is really interesting is this light fusion, this light sensor protein, which is a fusion of CPH1 and NZ that's used for the bacterial photography system. And if you download the sequence for CPH8 and put it into a design tool made by Ginkgo, what it will tell you is that in fact CPH8 has a PST site in the middle of it. So what is the strategy then, right? So now we have a piece of DNA that we think is really interesting. We'd like to make a standard biological part of it, but within that sequence there is one of these restriction sites that we need for cloning. If we look at the entry for CPH8, this CPH1-NZ fusion, the entry for this part that's in the registry of standard biological parts, and we look at the design page, one thing that we'll note is that the people who entered this part into the registry put a silent mutation into the part at base 108. They changed a G to an A, and by doing that they removed the PST site from within CPH8 to enable it to become a standard biological part. Now this of course requires that that change, G to A, not change the amino acid sequence of the resulting protein, but because of the wobble position and the chance that you get with the degenerate genetic code, it is possible to make a change at the DNA level that will remove restriction sites that does not change the amino acid sequence of the final product, in this case a protein. So this would be an example of a standard biological part. The benefit, of course, to refining all these parts now is that it allows you to build them without having to come up with a unique strategy every single time you want to put two pieces of DNA together. And it takes the sort of guesswork out of assembling DNA. Of course, putting the pieces of DNA together is not the same thing as having them work reliably in the cell. So just because you can reliably compose DNA does not mean that it will be reliably functional. So on the left here we have a bolt and a nut, and if you tighten those together, we would be very surprised if those didn't work together well and do what they were supposed to do. By contrast, we have a DNA fragment and a plasmid that we're mixing on the right here, putting that piece of DNA into the plasmid backbone, putting them into cells and identifying clones. And it's really not obvious or not always known that the piece of DNA that you put into a recombinant plasmid is going to behave in the way you would like it to behave. So physical composition gets us part of the way. What we'd really like is functional composition of parts. And one strategy for generating functional or for reliably functionally composing parts might be to rely on data and measurement in order to know how to use them. So on the left here is a data sheet for a phototransistor. It has a lot of specifications about how that part should be used. It talks about, under what conditions to use it, when it works best, when it might break, things like that. You might also see on the right here a data sheet for a biological part. Now this is a first effort at trying to define the parameters for using a biological part so that you might be able to use it reliably. In this case it has compatibility and reliability, when to use it, when it might break, what it looks like when it breaks. And so one way perhaps to get at functional composition of parts would be to define these use cases for all of the parts that we would want to compose with, and use the parts only in ways that have been defined and that we can know it works. So the challenge here, one of the great challenges here, is to generate sufficient amounts of data and sufficient amounts of useful data so that people can look at these sheets and can rely on them and would actually decide to build a complex system based on what they've read by the data specification sheets. And so because so much data needs to be measured, and determined, compared from lab to lab, we'd like the measurements we make here at MIT to be different, or to be the same, not different, from the measurements that you would make in UC Berkeley or Purdue or any of the schools in between. Any high school in the nation should be able to make some measurements, know how they compare to the measurements that are being made in other places, and use the parts reliably. So I'll encourage you to watch the next lesson in this series about standardization and measurement. It's a video from my friend Jason Kelly, who's at Ginkgo Bioworks, talking about a measurement kit that will enable promoters to be standardized in the measurements of promoters to be standardized and compared from place to place. And it's an interesting one, refers back to the physical reference standard of the ohm and things like that. So I'll encourage you to watch that. This ends the quick lesson here, though, the introduction to standardization and measurement. I hope it's been helpful, and I hope you'll continue to enjoy the BioBuilder work.